an important long answer or important question from the excretory system in human being, that is nothing but urine formation. What are the various steps that are there in urine formation? Let us have a glance on it. Let us take the various steps, whatever involved in it, and how urine is formed in human beings. Let us discuss now. Let us take urine formation and various steps. There are basically, it is completed by various stages. There are three major stages are involved in the formation of urine. The first stage is ultrafiltration, nothing but glomerular filtration. And the second step is, or second stage is selective reabsorption. And the third stage is tubular secretion. Now let us learn about ultrafiltration. What do you mean by ultrafiltration? Let us learn about ultrafiltration. Here, ultrafiltration occurs inside the glomerular capillaries. What are they? Glomerular tubules or capillaries that are there inside the Bowman's capsule of the nephron. If you take the Bowman's capsule here, Bowman's capsule contain glomerular tubules that are formed by efferent and efferent arterioles. Now here these two together makes a fine microtubules called the glomerular tubules inside the Bowman's capsule of a nephron. Now among these two diagrams, first diagram is necessary and you have to draw the rough sketch or rough diagram you need to draw in the final examinations. Whereas if you take the second diagram, it is not necessary. Now, what is glomerular filtration? Let us see. When the blood enters into the efferent arteriole, you know very well about efferent arterioles. These efferent arterioles are smaller blood vessels that are entering into the Bowman's capsule of the nephron. Now, when the blood flows inside this efferent arteriole, it has some hydrostatic pressure. That is totally 60 mmHg. Now, this hydrostatic pressure, when the blood enters into the capillaries, it occurs. The hydrostatic pressure, nothing but a 60 mmHg, it is opposed or objected by colloidal osmotic pressure of the proteins in the glomerular tubules. It is about 32 mmHg, whereas Bowman's capsular pressure, basically Bowman's capsular pressure contain hydrostatic pressure about 18 mmHg. So this 60 mmHg total pressure is opposed by 32 mmHg, nothing but colloidal osmotic pressure of the proteins, and hydrostatic pressure, Bowman's capsular hydrostatic pressure. So hence 32 minus 18, that is nothing but 10 mmHg is the net filtration pressure in the glomerular tubules, which plays an important role in the filtration process. So this causes filtration of blood through three basic layers. These layers are formed by endothelial cells of the glomerular capillaries. Now, blood is filtered through fine microscopic pores called slit pores and also apertures. Commonly in the textbook or material it is mentioned as fenestration, nothing but apertures or slit pores. So these slit pores are able to filter, or they are able to filter because of specific net filtration pressure. Now, therefore, this process, which is commonly called as a, or scientifically called as a ultrafiltration pressure. Now, second step in the process of ultrafiltration and urine formation is selective reabsorption and secretion. So the tubular epithelial cells in different segments of the nephron plays an important role in the selective reabsorption and secretion. They are able to reabsorb various substances or materials of the glomerular filtrate. by active or passive transport. About approximately 85% of the filtrate is reabsorbed in the PCT itself and the descending limb of loop of Henle. Now, based on the necessity of reabsorption, the substances of a glomerular filtrate classified into various substances. First one, high threshold substances which are highly reabsorbed, such as like glucose, amino acids, vitamins, and some salts. Proteins cannot be reabsorbed. Why? Because they are larger in structure. Structurally, they are large. And low threshold substances which are absorbed in a little amount. That's why they are called low threshold substances. Low means little. Example, urea and uric acid. And finally, A threshold substances which cannot be reabsorbed. Example, creatinine. What is that? Creatinine. Finally, in the PCT, what happens? Let us see. What do you mean by PCT? Let me show you in the diagram. No need to draw the diagram once again, but for your understanding, I am drawing this diagram. You can see here. So this is the region, first convoluted tubule called proximal convoluted tubule. This proximal convoluted tubule basically contain a finger-like structures inside their tubular structures. These finger-like structures are called microvilli. The microvilli specifically increases the surface area of absorption. Increases the surface area of absorption. All the essential nutrients, whatever the essential nutrients are there, about 70 to 80% of the electrolytes and water are reabsorbed by this segment. That is very important point. Sodium, if you take the sodium, is actually transported into the cortical interstitial fluid. The positive charge movement causes passive transport of chlorine. If you suppose take a positive charges, the positive charge movement causes passive transportation of chlorine. Now, finally, glucose, if you take the glucose and amino acid and other essential substances are actively transported. They are actively transported. They are transported by the active process. Movement of water occurs by osmosis. You know very well about osmosis, right? Movement of water occurs by osmosis. Now, in the Henle's loop, what happens? Reabsorption is minimum in this segment. Reabsorption is very minimum in this segment. Now, finally, it maintains high osmolarity of the medullary interstitial fluid. The descending limb is permeable to water. If you take the descending limb of loop of Henle, it is permeable to water but almost impermeable to electrolytes. Whereas if you see, because of this character, as a result, the filtrate concentration is gradually increases when it moves towards the inner medulla. Now, ascending limb, opposite to this limb, ascending limb is there, which is impermeable to water. Hence, filtrate becomes progressively more dilute due to presence of electrolytes. Finally, DCT. How DCT plays an important role in urine formation. Cells are very shorter in the DCT region. Hence, in this region, cells cannot form finger-like extensions called microvilli. Due to absence of microvilli in the DCT, they are not involved in reabsorption. Now, conditional reabsorption. But what type of reabsorption occurs in DCT? Facultative reabsorption or conditional reabsorption is occurring. What do you mean by that? The facultative reabsorption occurs by any type of chemical or hormone, etc. So the hormone which is involved in the facultative reabsorption in DCT is nothing but here the ADH hormone, anti-diuretic hormone, nothing but vasopressin. This helps to reabsorption of water in this segment. Now, and selective secretion of hydrogen ions, K plus ions, and NH4 plus ions, NH3 plus ions into the DCT from peritubular capillaries also occurs in this region. Now, apart from this, in the collecting duct always carries what it carries, filtrate through the medulla to the renal pelvis. Considerable amount of water is reabsorbed in this segment. Apart from this, it plays an important role in the maintenance of pH and ionic concentration of the blood by various various substances. So this is for the end of the session in the collecting tube and collecting duct, how reabsorption occurs we have seen. This is enough for long answer, and don't forget to subscribe and after watching this video. We'll see in the next video once again. Thank you.